Hello, welcome all of you to my presentation. I'm Dr. Sanger and I'm going to present to you lecture 1 of session 2, which will be about membrane permeability. And Dr. Daria will present to you lecture 2 of the current session, which will be about the ATP-dependent pump and exchangers. So I wrote my email in the first slide, and please send me an email about anything that you couldn't understand it very well in my lecture, and I will be more than happy to reply to your email as soon as possible. So, as I'm aware that you got in your first session membrane structure and membrane dynamic, and today I'm going to talk about the membrane permeability. So our aim of this lecture is to give you the idea about the major transport mechanisms that contribute mainly to generate different ionic gradient across the cell membrane, and also to give you an idea how different substances use different type of the transport mechanism across the cell membrane, depending on the characteristic of these molecules. For example, how water-soluble molecules could be moved on either side of the plasma membrane, and how our cells could extrude our metabolic waste product to outside of the cell. And also, we need to know the impact of these different transport mechanisms and different cellular physiological function. And hopefully by the end of this lecture you will be able to learn how different molecules could move across the cell membrane, and you will get an idea also about what is a diffusion, what is a simple diffusion, what is a facilitated diffusion, and how you could distinguish diffusion from active transport. And also you will get a bit more details about channel proteins. So, starting with our today lecture, all of you, I believe that you have an idea about different ionic composition on either side of the plasma membrane. For example, the composition of the intracellular fluid is different from the composition of the extracellular fluid for different solutes. Let's say, for example, the extracellular fluid contain large amount of sodium in comparison to inside of the cell, while the intracellular fluid contain large amount of the potassium in compared to outside of the cell. And we need to keep these different ionic concentration on either side of the plasma membrane in such a way, and important to keep our cells integrity. So in this lecture you will get an idea how this ionic composition gradient could maintained. So from your previous lecture, I believe that you got an idea how the cell membrane, or how the plasma membrane are structured, and the composition of our plasma membrane. For example, you know that plasma membrane consists of phospholipid, proteins, cholesterol, and carbohydrates. And you know that the phospholipid part of our plasma membrane arrange themselves in a way that give you a double bilayer, in a way that head of phospholipid, which is a phosphate, face the water component of extracellular compartment and also the inner portion of the head of the phosphate facing the water component of intracellular compartment, while the hydrophobic tail face each other. And we know from your previous session that we have proteins embedded on the plasma membrane, and this protein could be integral or could be peripheral, and the integral protein could act as a transport protein. Such transport protein could be like a channel protein or could be a carrier proteins. And we know that lipid-soluble substances could be passed very easily from either side of the plasma membrane. For example, oxygen, carbon dioxide, alcohol could be moved freely without any barrier, while water and water-soluble substances could not be passed through the lipid bilayer. Especially they will get stuck in the hydrophobic tail of the fatty acid on the plasma membrane. So in this lecture we are going to talk about how water and water-soluble substances could be moved on either side of the plasma membrane, and what are the role of transport proteins to achieve this process. So as I mentioned that water-soluble substances or charged molecules or large molecules could not be passed through the lipid bilayer. So how they could move? For example, charged molecules like ions like sodium, potassium, chloride, calcium, how they could move? Or large molecules like glucose or amino acid, how they could move? So to move these molecules we need help from proteins embedded on plasma membrane. These proteins could act as a channel protein. They could arrange themselves in a way that they give you a tunnel passageway, a watery passageway for movement of charged molecules like sodium, potassium, calcium, chloride. And we have also another type of the protein called carrier protein. This carrier protein, they have a receptor for binding site to different water-soluble molecules that later on could do shape changes to bring these molecules inside or outside of the cell. So if you look to this slide, you could see that we have a double bilayer of phospholipid, the phosphate head facing the watery portion of extracellular and intracellular compartment, and the hydrophobic tail of the fatty acid face each others. And you could see that lipid-soluble substances like oxygen, carbon dioxide, nitrogen, they could pass easily across the plasma membrane, while water-soluble substances such as glucose or amino acid or charged molecules could not be passed. They need help from proteins embedded on plasma membrane. Water is a special situation. They are quite small enough to be passed also through the specialized channel protein called aquaporine. Aquaporine channel available on our plasma membrane that give the water a passageway to be moved on either side of the plasma membrane. So, going to talk about the process of membrane transport, we need to know we have two types of membrane transport. We have passive membrane transport and we have active membrane transport. The passive one is also could be termed diffusion. As a diffusion, we mean the movement of molecules from the area of high concentration to area of low concentration. And this process does not require energy. It is mainly means movement of ions or other substances across the membrane in a way that could be through the simple diffusion without any help for lipid-soluble substances, or could be through the combination with a carrier protein, or passes through the channel protein from the area of high concentration to the area of low concentration. And it usually does not require energy. And the energy that causes diffusion is the energy from normal kinetic motion of matter. And we could subclassify this mechanism into simple diffusion and facilitated diffusion and osmosis. And we are going to cover all of these subtypes in the next few slides. The second type of membrane transport is active transport. And the difference between the diffusion or passive membrane transport and active membrane transport is that in active transport, molecules are moving from the area of low concentration to the area of high concentration. And this process requires energy in form of ATP. And so this process requires specialized pumps or specialized carrier protein that help this process to be achieved. And this process could be subdivided also into primary active transport or secondary active transport. And in the next few slides, we will talk about the difference between these two types. So, why we need a membrane transport? Generally, we need membrane transport to maintain intracellular pH. And hopefully in next few sessions you will learn how the proton, how hydrogen ion would be moved from the intracellular space to the extracellular space, or from extracellular space to the intracellular space, in a way that to keep our intracellular pH in the normal range. And also membrane transport allows our cells to keep the ionic composition on either side of the plasma membrane, which is very important for nerve impulse, for example. And membrane transport is also important to regulate cell volume. And when you talk about the osmosis and sodium-potassium pump, you will get an idea how this process is important to keep our cell volume in a healthy state. And also we need to bring some metabolically active required material like glucose, amino acid to the inside of the cell, and to extrude other waste product to the outside. And such process of the membrane transport is very important to give you the ionic gradients that we require for excitable cells like nerves and muscle to initiate an action. So, starting with the passive membrane transport. The passive membrane transport, as we said, it is a process of movement of molecules from the area of high concentration to the area of low concentration. And this process does not require energy. And this process of the movement of molecules could be from the area of high concentration to low concentration, or could be from the area of high pressure to the low pressure, or we could depend on electrical gradient. And this process will be continuous until we reach equilibrium. We will get, for example, the process of transport of any molecules will not be stopped until we will get equal distribution of the concentration of specific molecule on either side of the plasma membrane. So, if we are going to talk about the concentration gradient, usually we could the molecules could be moved down gradient, so from the area of high to low, which is true for passive membrane transport, or in opposite direction, which is from the area of low concentration to the area of high concentration, which is true for active membrane transport. While for electrical gradient, we need to know that usually it's very important that to know that inside our cell we have proteins, we have organic molecules that are negatively charged that could not escape to outside of the cell, and that give you more negativity inside the cell, and the outside usually you get more positivity. In the next few sessions, you will learn how this process of different membrane potential or different electrical potential is very important for the nerve impulse and muscle contraction. And usually, the positive charge attracts the negative charges, while negative charges repel negative charges. So that is the process of the movement of electrical gradient. So, starting with the passive membrane transport, we could subclassify this transport mechanism into 4 major types. The first one is a simple diffusion through the lipid bilayer. Second one is through the protein layer, which is mainly when we are meaning channel protein, and we have facilitated or carrier mediated diffusion and osmosis. So in the next few slides we are going to cover all of these different types of passive membrane transport. So regarding the first type, that's usually for lipid soluble substances. For example, oxygen, carbon dioxide, alcohol, they are moved from the area of high concentration to the area of low concentration through the lipid bilayer without any help. That's why they are simple. And it's very important to know that the rate of the diffusion for these molecules is actually depending on lipid solubility of the substances. For example, large amount of oxygen could be transported in that way, and because they are largely soluble, largely lipid soluble, they could be delivered into the interior of the cell in a way that the cell membrane did not exist. So that is a process of that's the definition of the simple diffusion through the lipid bilayer, which is mainly true for lipid soluble substances. The second type, usually we mean is movement of charged molecules through the protein channel. So within the plasma membrane, we have proteins. These proteins actually account for more than 50% of the plasma membrane composition. They could be available either in the form of integral, which is penetrated all the way through the plasma membrane, or presented as a peripheral protein. Some integral protein within the plasma membrane, the amino acid composition of this protein, they arrange it themselves in a way that they give you a watery passageway penetrated all the way through the plasma membrane. So this specialized structure or form of the integral protein called channel protein, and this channel protein, depending on the size and also electrical charges of the amino acid composition of the protein, they could be very selective for each of the specific charged molecules or ions. And these channels could be continuously opened, that we term it leaky ion channels, which are always open, or could be gated, so they are closed and could be opened on a specific demand. So such gated could be also subclassified, and we talked about it in next slides. In addition, we have also specialized protein channel for water called aquaporin water channel. That's also a channel protein that allows a free passage of the water molecules across the plasma membrane. So if you are going to take a look at this picture, we could see the double bilayer of the phospholipid, and we could see that the integral protein, they could arrange the amino acid composition of such integral protein could arrange themselves in a tetrameric structure that could give you such central pore that allows the free passage of ions across the plasma membrane. So depending on the size and shape and electrical charge of the amino acid, they could allow specific ion to be passed and not other ion to be passed. And this property of the channel proteins called selectivity. So if you want to review some of this information in this diagram, we could say that we have a double bilayer of the phospholipid, which act as a barrier for water and water soluble substances. And within the plasma membrane, we have integral proteins, and this integral protein could act as a receptor, or could act as enzymes, or could act as a protein channel, or could act as a carrier proteins. And we have a carbohydrates also, and we have also cholesterol for the stability of the plasma membrane. So molecules which are lipid soluble, they could be easily transported across the plasma membrane. And water soluble molecules, they require additional help from embedded proteins on the plasma membrane. This could be in the form of a channel, watery passageway, penetrated all the way through the plasma membrane for ions and water, or could be in the form of carrier proteins. So, as I explained it, the selectivity or specificity of the ion channel, we need to know that potassium channels are only permeable to the potassium ions. This is not 100% true, because it could allow the passage of the sodium molecules also. But for example, if you have in one specific set of time 100 molecules, 99 of these 100 molecules are potassium and 100 molecules of sodium, such specific potassium channel would allow more potassium ions to go inside of the cell. To go to outside of the cell, because these channels is the example of passive transport. So it would be from the area of high concentration to the area of low concentration, and potassium concentration inside the cell are higher outside the cell than outside the cell. So if these channels are open, they will allow the passage of the potassium from outside to the inside. And you have also sodium channel, we have also calcium channel, we have also chloride channel. So these channels are mainly permeable to the ion that named these channels. For example, you have sodium channels, which allows the passage of sodium ions. And if these channels are leaky, that's continuously open, when they allow the movement of the sodium from the outside to inside, because the concentration of the sodium is higher outside than inside. And that's the passive channel protein are example of the passive membrane transport. So, as I mentioned in some slides before, a few slides before, these channel proteins could be continuously opened. It's called leaky channels. They are not gated. While we have other channel proteins that are gated, they are closed in normal resting situation, but could be open on a specific response. These are called gated channel. And we could subclassify gated channel also into voltage gated channel or ligand-gated channels. So for voltage-gated channel, we need to know that difference of electrical membrane potential could allow such voltage-gated channel to be opened or closed. For example, if we are going to talk about sodium channel, gated sodium channel, voltage-gated sodium channel, the gate for that channel actually located on outer surface of cell membrane, and when the inside of the cell get more positivity, the gate for the sodium channel will be opened. And this is an example of voltage gated sodium channel, which is responsible for the nerve impulse. So any change in electrical membrane potential could also change the gate of the voltage gated channel. However, the second type, which is called ligand gated channels, it means that such protein channels on the plasma membrane have a receptor. Such receptor are specific for specific ligand. When we have specific ligand bind to such receptor on the ion channel, there will be shape change of that ion channel and lead to the opening of such ligand gated channel. And the important example of ligand gated channel are acetylcholine channel. So we have this channel on the plasma membrane, and these channels are usually closed, but they have a receptor for acetylcholine neurotransmitter. When such acetylcholine bind to that receptor located on that channel, lead to the shape change and channel opening later on, which is very important for the nerve impulse and muscle contraction. Gap junction is another example of specific type of the channel which are located between adjacent muscle cells, for example, and cardiac muscle cell, and other types of the tissue. So regarding the voltage gated channel and ligand gated channel, you will get more information on how these two channels could be open or closed in more details in next few sessions. The third example of passive membrane transport is through the facilitated diffusion or mediated diffusion. So we have some molecules like glucose or amino acid. These molecules need to move from the area of high concentration to the area of low concentration. But because these molecules are large, could not be passed through the plasma membrane, and they need additional help from the proteins embedded on plasma membrane. So we have carrier proteins. These carrier proteins actually got a receptor binding site for glucose or amino acid. And when we get a binding between such molecules, the receptor binding site on the carrier protein, we will get shape changes and we will bring such molecules to inside of the cell. If you take a look at this image also, we could see that we have a carrier protein embedded on plasma membrane. Such carrier protein got a receptor binding site for transported molecules, for example, glucose or amino acid. When such transported molecule bind to the binding site on the carrier protein, we will get a shape change, and we will allow the movement of that transported molecule to inside of the cell. This process is an example of passive transport mechanism, which is from the area of high concentration to the area of low concentration, and this shape change does not require any energy in form of ATP. So another example, which is another picture for facilitated diffusion. For example, we have glucose outside of the cell, and we need to move this glucose from the area of high concentration to the area of low concentration. So we need carrier protein on the plasma membrane. So such carrier protein for glucose we call GLUT, GLUT transporter carrier protein. And this transporter protein available widely on our cells, and it allows the movement of glucose from high concentration to the area of low concentration. And some of these glucose are insulin sensitive. So in those patients who doesn't have enough amount of insulin, or they don't have insulin at all, these glucose transporter, which are insulin sensitive, they could not help, they will not be activated to allow the passage of the glucose from outside to the inside. So the glucose will stay in the circulation, and thus it gives you rise in blood sugar, and then later on diabetic condition. The final example, the final type of passive membrane transport is osmosis. So we talked about water movement across the plasma membrane, and we said that water could not be moved freely like oxygen across the plasma membrane, and they require specialized protein channel called aquaporine water channel. And we have another term called osmosis, which is the movement of water from the area of low concentration of solute to the area of high concentration of solute. So what are the solutes? Solute could be any molecules inside the solution, like glucose, amino acid, sodium, potassium, and the solution usually made of such solute and solvent, as a solvent could be water. If you take a look at this picture, we could say that we have a semi-permeable membrane that are only, for example, in this picture, permeable to water. And on compartment A, we have 3 solids, and in compartment B, we have more solids. So the water will be moved from the area of low solute to the area of high solute, or from the area of low concentration of water, from the area of or from the area of high concentration of water to the area of low concentration of water. This is the process of osmosis. For example, the concentration of water here, the volume of the water here in A is more than B, so it will be moved from the high concentration of water to the low concentration of water, or from the area of low solid concentration to the area of high solid concentration. This process is very important to keep cell volume, and they are very important when you talk about the process of water follow solute always. For example, in the process of renal reabsorption or intestinal absorption of solutes, you need to know always water follow solutes. So we finished the passive membrane transport, and we are going to talk about the second type of the membrane transport, which is active transport. So the active transport, it's actually the process of movement of molecules against concentration gradient. So from the area of low concentration to the area of high concentration, which is the uphill transport. This process requires adenosine triphosphate, require ATP in the form of high energy phosphate bonds. So if you want to give you a difference between active transport and facilitated diffusion, we need to know first that in active transport we have the movement of molecules against concentration gradient, while in facilitated diffusion They are moved from the with a gradient, not against gradient. And in facilitated diffusion, we do not require energy, while in active transport we require energy. So that's a major differences between the active transport and facilitated diffusion. In both of these type of transport, we require carrier protein, but the difference is the way that the molecules are moving and the way that they are moving, does they require energy or not. So, substances which are transported actively could be in ionic form or could be non-ionic form. For example, ionic form like sodium, potassium, calcium, hydrogen, non-ionic form like glucose, amines. As we mentioned that active transport will be subdivided into primary active transport and secondary active transport. The primary active transport is a transport of molecules from the area of low concentration to the area of high concentration, and this process requires energy in form of breakdown of ATP. While in secondary active transport, we need to get established a gradient for such ionic or non-ionic form. Such established gradient will be achieved from primary active transport, and the energy, the storehouse energy that was used from the primary active transport to establish such gradient will be used for secondary active transport. So we need first to have primary active transport, and such primary active transport will give you gradient for specific ion or non-ionic form. After that, the secondary active transport will use such gradient to achieve specific type of membrane transport mechanism. This process is called secondary active transport. And secondary active transport will not be happened if you don't have a primary active transport. That's why we term it secondary active transport. The main and the most studied example of primary active transport is sodium potassium pump. Sodium potassium pump is a very important pump. It is an example of primary active transport. It means that moves sodium and potassium against concentration gradient. And because it is an example of primary active transport, it requires energy in form of ATP. So, if we are taking a look at this picture, we could see we have a phospholipid bilayer and we have this pump, which is a sodium potassium pump. So if I am going to move sodium in active transport, it means that I am moving sodium from the inside to the outside, because inside sodium concentration is lower than outside. And if I am going to move potassium in any example of active transport mechanism, it should be from the outside to inside, because concentration of the potassium outside is lower than the concentration of potassium inside. So that's why they called active transport. So this pump, sodium potassium ATPase pump, it moves 3 sodium from inside of the cell to the outside, and it brings 2 potassium from outside to inside, and this process requires ATP, breakdown of ATP to the ADP, and use the energy of the phosphate bond to achieve this transport. So it's very important and it's very crucial to not forget this information. NAK pump moves 3 sodium from inside of the cell to outside of the cell, and it brings 2 potassium from outside to inside of the cell, and requires ATPase enzyme to break down ATP to ADP as a phosphate. So why we need NAK pump? If you think about NAK pump, you need to know that at the end of the action of this pump, you will get one more positive ion to be lost inside the cell, because we are moving 3 sodiums to outside, which are positively charged, and we are bringing instead 2 potassium, which are also positively charged, to inside of the cell. So at the net result of this pump, you will lose 1 positively charged ion inside of the cell, and this gives you a negative membrane potential inside of the cell, which are very crucial for electrical membrane potential and nerve impulse. That you will get all of these different information about resting membrane potential, electrical membrane potential, depolarization and repolarization in next few sessions. So NAK pump is electrogenic pump, because at the end of the action of this pump, you will lose 1 positively charged molecule inside the cell. And also, this pump is very important to maintain cell volume. In next slide, you will learn how such pump could maintain cell volume from rupture. So, with the action of this pump, we are protecting our cells from getting swelling and later on from being ruptured. How? Inside our cell, as I mentioned before, we have proteins, we have other organic molecules that could not be escaped from the cell to the outside, and these proteins are negatively charged, so they attract positively charged ions like potassium, sodium, and other positive ions. So in previous few slides we talked about osmosis, and we said in osmosis water follows solids. So if proteins inside the cell attracting solids like positively charged solid like sodium and potassium, and water follow them to inside of the cell, our cells will be swollen and later on will be burst. However, we have NAK pump on our plasma membrane. Such NAK pump will take 3 sodiums outside of the cell and bring 2 potassium inside of the cell, and this process, it gives you always it takes 3 solids to outside and bring 2 solid inside, and this process, it gives you always a negative loss of solid inside of the cell. So we need to know the end action of the NAK pump is to keep cell volume and to provide a negative membrane potential inside of the cell, which are very important for the nerve impulse. Secondary active transport, it is an example of active transport, and it means that you have primary active transport, which operating in a way that give you specific gradient for specific ion. Such gradient could be reused for secondary active transport. For example, we have sodium and potassium primary active transport pump. It gives you more sodium outside and also more potassium inside of the cell. Such gradient that we got at the end of the NAK pump action, it provides you a gradient for sodium, that the outside, the concentration of sodium will be higher than the inside. So the sodium will have a tendency to come back to inside of the cell. Such sodium, when they tend to come back to inside of the cell, will bring another molecules. Such carrier protein that bring sodium from the area of high concentration to the area of low concentration, that such gradient established from the primary active transport and bring another molecules against concentration gradient from the area of low concentration to the area of high concentration. This is an example of secondary active transport. If the direction of movement of second molecule, the sodium or potassium the first molecule, the same direction is called symport. If they are in the opposite direction, they called antiport. So next few slides, we are going to explain this example more. So take a look at this picture. We have NAK pump initially, which is a primary active transport. Such NAK pump provide you a gradient for sodium. Such gradient will be from outside to inside because it brings 3 sodium to outside. So we have carrier protein embedded on the plasma membrane. These carrier protein embedded on the plasma membrane, they have a receptor binding site for sodium and another molecule, for example, glucose. So the sodium tends to come to inside of the cell and they bind to such carrier protein. But because this carrier protein is an example of secondary active transport, they need to bring an additional molecules against concentration gradient. For example, here is a glucose. So they will bind to sodium but will not bring sodium to inside, like an example of carrier protein of facilitated diffusion. But it needs to bind also with a glucose molecule. After binding to the glucose and sodium, they will bring both molecules. They will do conformational or shape changes and then bring sodium and glucose to inside of the cell. So the movement of the glucose will be from the area of low concentration to the area of high concentration. That's why they called active transport, which is secondary. Why secondary? Because they used the gradient that established from the primary active transport for sodium. And that's also the same for amino acids, the same direction. However, it could be in opposite direction. For example, it could bring sodium to inside and exchange it with calcium, and that's very important to maintain the intracellular calcium concentration at a very low concentration. And also, it could exchange the sodium with a proton. That's very important to maintain the intracellular pH also. These are the example of antiporter, secondary active transport. So mechanically, when you are going to talk about this type of active transport, secondary active transport, we need initially to have operated primary active transport to get a storehouse of the energy that was used in primary active transport. At the end of the primary active transport, you will get a gradient for specific ion. For example, in NAK pump, you will get a gradient for sodium and potassium. So you will get a gradient for sodium to be higher at outside from the inside. So they will tend to move back to inside of the cell through the carrier protein. But this carrier protein got 2 binding sites, one for the sodium and one for another molecule like glucose or amino acid, if they moved in the same direction, or they got a binding site for calcium or proton if they moved against in opposite direction to the sodium. In this example, we are talking about the co-transport, which will be in the same direction. So sodium that will get such gradient for sodium from primary active transport bind to carrier protein, but it will not be bringing sodium to inside until we will get binding to the glucose also. After that, we will get a conformational shape change and it will bring both of them to inside of the cell. This process requires initially operated primary active transport. And this process does not require energy because the energy that has been liberated from the primary active transport to be act as a storehouse for such gradient, and it brings another molecule against gradient, which is, for example, here glucose from the area of low concentration to the area of high concentration. So glucose, amino acid, could be moved from the area of high concentration to the area of low concentration using facilitated carrier diffusion, or could be moved from the area of low concentration to the area of high concentration using sodium glucose cotransporter. For exchanger, here we have an opposite direction of movement of sodium, for example, exchange with the calcium, or sodium exchange with proton. and this process is a secondary active transport. They require initially operated primary active transport to give you a gradient, for example, for sodium, and after that such carrier protein bind to the sodium and will not be moved until we will get also binding of the calcium from inside. Why from the inside? Because this is a secondary active transport, so the second molecule should be moved against concentration gradient, and the concentration of the calcium inside of the cell is lower than the outside, so they are moved against concentration gradient. In this picture, I will try to explain how secondary active transport it requires operated primary active transport. So these are the lumen of kidney, or could be the lumen of the intestine. We have a brush border epithelial cells, and we have interstitial space and circulation. So here, this membrane, this part of the membrane called basolateral membrane, and here is called apical membrane. And on the basolateral membrane, we have a primary active transport for sodium, potassium, ATPase pump. This pump, what are the action of these pumps? It takes 3 sodium from inside to the outside, and it brings 2 potassium from outside to the inside of the cell. So the concentration of the sodium inside of the cell will be lower than the inside of lumen of the nephron or intestine. So here sodium tend to come back to inside of the cell because of the gradient that we established from the primary active transport. But this sodium will not be moved through the channel. They will be moved through this carrier protein. This carrier protein will be an example of secondary active transport. It could be sym-porter or it could be antiporter. If we are going to talk about the example of sodium glucose co-transporter, so this carrier protein will have a binding site for the sodium and will have a binding site for the glucose. But here the glucose will be moved from area of low concentration to the area of high concentration. If we don't have such pump and such sodium gradient, this pump will not be active, and we will not take a glucose from area of low concentration to the area of high concentration. And after that, the concentration of the glucose here will be increased, and it will be transported to the circulation through the facilitated diffusion or glucose transporter facilitated carrier diffusion. I hope this picture simplify the cooperation between the primary active transport and secondary active transport. This picture will give you the summary of what we have talked about today lecture in terms of major important points that need to remember for each type of different transport mechanism. For example, for simple diffusion, you'll get the movement of the molecules downhill. Do we need any carrier protein? No. Do we need any energy? No. Do we need any primary transport gradient for sodium or potassium? No. For example, in facilitated diffusion is similar, but here we require carrier protein, but still we don't require any energy or any gradient for sodium or operated primary transport. While in primary transport, it will be against concentration gradient, and it will require energy, okay, and it will require specific pump or carrier protein. It could be co-transporter or could be counter-transporter. These are the example of secondary active transport. And for these two example of secondary active transport, we require gradient, sodium gradient, or initially operated primary active transport. I hope this lecture was useful for all of you, and in next few sessions you'll get a better idea on electrical gradient, on membrane potential, and also on the process of voltage gated ion channel, ligand gated ion channel. And please, if you have any question, email me and I'll try to explain to you your question, I could provide you more images or links of some videos that could solve your problem. All the best for all of you.